We'll find out.
Okay,
Right?
so let me introduce our next group. So this is group number 5, who will present. I don't know why I'm having such trouble pronouncing the titles of people's groups. So the last one I really want you to. These are very easy. So erythros.
Erythros. Right.
Erythros.
Yeah.
As in a red blood cell.
Correct.
Technologies. So you can see Kevin, Taylor, Shelby, and Danica will present.
All right, thank you for the introduction. So we're Erythros Technologies. We'll be presenting the final design report for the production of enzymatically converted type O blood. Here's Taylor, Danica, Kevin, and I'm Shelby. To start off, the topics we're going to cover today are first introduction regarding some of the science and the research behind our process. And then we'll talk about a detailed process description, economics analysis, and we'll conclude with regulatory and safety considerations. Each day in the United States, approximately 3 transfusions are made in error, and this refers to when a person receives the wrong blood type. This can be very dangerous and can cause kidney failure, strokes as a result of extensive blood clotting, and in some cases can be fatal. But type O negative blood is a universal donor because it's accepted in all blood transfusions. However, only 8% of the US population has type O blood. Here's a chart describing the contents of the different blood types. So there's type A, B, AB, and O. So for example, if a person who has type A blood receives a B transfusion, the antibodies in their plasma will attack the B antigens on the foreign blood, and this is what elicits the immune response. However, type O blood does not contain the A or B antigen, so the antibodies in the patient's blood will not attack the O blood.
So obviously having like type O blood to always transfuse would be really useful, and research has been done to try to make O blood, and there's a lot of methods out there. So there's PEG masking, which kind of just masks the antigens. Then you can do an enzymatic conversion where you're clipping the antigens. And then you can get way more into it, and they're looking at like stem cells, modifying stem cells, and even genetic engineering. We're going to focus on the first 2 for this presentation, which are PEG masking and enzymatic conversion.
Okay, so as Taylor said, one of the first methods we're going to look at is the enzymatic conversion of antigens. So just to give a brief overview of how that actually works. The antigens have a specific binding site where the antibodies will attack. So basically what these enzymes can do is clip the site where the antibody binding occurs, and that basically renders the antigen inactive. So when that binding doesn't occur, the immune response doesn't happen, making the blood essentially behave as type O. So you can transfuse it without that immune response because of the deactivated form of antigens. So we're going to focus on one particular enzyme. It's been discovered where one single enzyme can convert both type A and B blood. So it was first isolated in 2005 and named to the GH98 family. That was the first member of that family of endobeta-galactosidase. So it was endobeta-galactosidase has been named EA base, basically indicating its specificity towards A and B antigens. It's been naturally located and also can be expressed recombinantly, which the recombinant method is much more commonly used just to generate more specific levels of the enzyme. So basically how this one works, it can cleave all of the B antigens, but because A antigens are slightly different, there are 4 different types of linkers on the antigens, so the cleaving is a little bit more complicated. So when the enzyme was originally discovered, it was unable to cleave all 4 types of A antigens. So recently, a paper published in 2015 came out that basically they took EA base and structurally evolutionized it so that it would have an increased ability to cleave those A antigens. They're basically calling these mutant type enzymes or mutant type EA base, which does have improved affinity towards the conversion reaction. So this slide refers to the originally isolated enzyme The figure 4 on the left shows where the actual cleavage happens on this is a AB type red blood cell, so it has both antigens. And as you can see, can I get this to work? Oh yeah. So this is where the linker is broken on the B type antigens, and this is where it's broken on the A type antigens. So that makes the red blood cell inactive. It's no longer able to bind to antibodies. So this chart on the right shows the evidence of EA base and its ability to convert red blood cells. So these plots here on the top show the histogram on the left is red blood cells only. This is A and B. The histogram on the right is A and B red blood cells in the presence of the antibody that it will react with. So when the antibody and the antigen bind, it has a change in fluorescence, which is where that peak moves over. So these two plots down here are show the red blood cells alone and also the converted red blood cells in the presence of the antibody. So as you can see, that antibody-antigen binding is no longer there, particularly in the B graph here. This is B blood cells. So the peaks completely overlap, showing that the binding is not there. It's not causing that shift in fluorescence. In the A, you can see there is a slight shift. So that's, like I said, it's due to the heterogeneity in the A linkers. So here is where the improvement was on the new enzymes. So this is a direct comparison between the original enzyme isolated in 2005 and the structurally evolutionized enzyme. So up here is just a control. The fluorescence is indicative of the antigen-antibody binding. Here is the no antibody control, so without that binding there is no fluorescence. In the quadrant 3 is the original enzyme. These are all A blood cells, by the way. So this shows the decrease, like I showed on that previous graph. It wasn't able to convert all of it, so you can still see some fluorescence. And then in quadrant 4 is the improved version of the enzyme. So we're showing much better efficiency. So moving forward with this project, we're going to focus on this evolutionized form of the enzyme to carry out our conversion.
So, like Danica just showed, you can't really get 100% conversion, so we didn't want to focus on just using the enzyme because we wanted to get as high as a conversion as we possibly could. So we're also using polyethylene glycol, also known as PEG, to mask our antigens, and this figure here shows how PEG works. It basically just forms a hydration sphere around the red blood cell, which if you have something blocking your antigens, then your antibodies can't bind. And to prove that PEG masked red blood cells actually didn't have a response when they were put in contact with their respective antibody, studies have been performed, and the results were assessed using flow cytometry. And this graph to the right shows that the antibody-antigen, the amount it got inhibited when you exposed your red blood cells to a certain dose of PEG. And as you can see from that figure, the maximum inhibition was when using 10 millimolars of PEG, which that is then the concentration that we decided to use for our process.
So our overall process goal is to be able to take type A, B, or AB blood and convert it to type O in a safe, reliable, and efficient reaction. We plan to achieve this with our novel process combining the two sequential reactions of the enzymatic conversion followed by the PEG masking. Another key aspect of our process is that all the tubing and bioreactor, basically any component of the reaction that comes in contact with the blood, is going to be single-use and disposable. This achieves a few things. It ensures the sterility of the blood, prevents against any contaminations, and it also reduces the upfront costs. Our goal is to produce 50 liters of blood per day with 5 10-liter batch reactions. We're also going to operate for 250 days a year with 2 shifts per day. Now, we're basing these numbers off a statistic that a hospital with a trauma center in Boston uses on average 500 liters of blood per day for transfusions, and so our goal is to meet 10% of this daily demand, or 50 liters.
Here's the product development pathway. So at this phase, we're intercepting the research and we're facing preclinical development. We project about a 5-year time span between now and when clinical trials will be completed, and at that point we will launch the product, which will involve manufacturing, sales, and marketing. followed by post-market support. And from preclinical development through post-market support, we'll be operating under design control to ensure product quality.
So like I said, we're going to operate in Boston, we're going to rent lab space, and provide blood directly to a single hospital. One of the benefits of doing this is there are 9 trauma centers in Boston, and so that shows us that there's a high demand of blood because we know there's a high patient volume in this city. Also, with all these trauma centers, we know that as we grow the company and are ready to expand, there's plenty of opportunity to do so from our single lab space. Hospitals will need our blood during times of blood shortages, which can occur for many different reasons, some being the harsh winter months, multi-vehicle collisions, or in a city like Boston with a heavy student population, the summer months when the city population drops when students are away. In terms of competition, there are no current companies that are commercially selling converted blood. Still though, the Red Cross and other major blood distributors pose a source of competition because they are able to sell blood, unconverted blood, for a significantly cheaper price point. And in fact, to run our process, we buy blood from the Red Cross and then perform our conversion and then turn around and sell it to hospitals. And so in terms of price point, it's hard to compete with that. In fact, we'll get more into that later, but our price is about 3 times that of the Red Cross. And this higher cost will get distributed between the patients, hospital, and insurance providers, and we believe it will be tolerated because it's going to be offered during emergent situations and provide life-saving interventions.
So to get into some of the specifics of the process itself, I'm going to go over the process flow diagram, details from each step of the process, as well as the processing equipment. So here's our process flow diagram. Like Kevin said, the entire thing is designed to be single-use and disposable. So each major piece of equipment, we have a bioreactor, a centrifuge, and a mixing tank. So each of those pieces will have basically a single-use bag inside. So at the end of each run, that can be disposed of. And each piece will be connected by sterile tubing and sterile welding. And the pumps will flow through the whole system. So we start with the reaction inside the wave bioreactor. The separation process happens in the centrifuge, and then the blood preparation and testing happens in the mixing tank before it's finally bagged as a product. So we have this process broken down into 6 major steps. The first 3 are detailed here. These are for the reaction itself, so the actual conversion of the blood. So we start with the batch preparation. Like I said, the reaction occurs in a wave bioreactor. It'll be 150 liters in size. So we start by adding 90 liters of PBS, followed by 10 liters of red blood cells, like Kevin said, raw blood coming from the Red Cross. So the reaction requires that the blood be diluted to 10% hemocrit, so 10 liters of blood in 100 liter working volume. After the addition is complete, we can start the rocking platform and make sure that the entire solution is well mixed before the reaction actually starts. So the second part is the enzyme conversion. So we'll add 14 liters of enzyme into the reactor. The enzyme is, based on the known activity of the enzyme, is that you need 14 units of enzyme to convert 1 milliliter of blood. So at a stock concentration of 10 units per milliliter, we'll need a total of 14 liters in the reactor. This reactor has a the enzymatic conversion reaction has a 60-minute reaction time. So after the 60 minutes, we'll reach the maximum level of conversion. And this reaction can happen at neutral pH and room temperature. So for this stage, because the reactions can all happen at room temperature, our plan is to control the actual temperature of the room rather than control from the bioreactor itself. So the temperature and the pH will be monitored but not actively controlled inside the system. So following the 60-minute reaction time, we have the PEG masking reaction. So this will take care of any possible unconverted antigens. So we have the polyethylene glycol is needed at 10 millimolar concentration inside the reaction mixture. So that comes out to 456 grams, which will be dissolved in 1 liter of PBS and then sterilely added to the bioreactor. This reaction also has a 60-minute incubation time to allow for maximum masking. So following this 60 minutes, the blood should be completely converted. we should have no more remaining antigens and the blood should have reached a safe level for transfusion. So now we'll move into the separation because you need the red blood cells to be pure to go back into the body. So we have a centrifugation step. Basically, red blood cells are much denser than anything else that's in the reaction mixture. So centrifuge works really well for the blood separation. The way that this works is the red blood cells or the reaction mixture from the wave bioreactor will be pumped into the centrifuge at a 3 liter per minute flow rate. The centrifuge actually works semi-continuously. So as it spins, the red blood cells are collected in a collection tank, and the waste is continuously removed from the top. So we can process a large volume without having to stop or use a bunch of small pieces. So as the mixing tank fills, the red blood cells will be pumped into the mixing tank, where the anticoagulant is added back. So anticoagulant is added at 1 part anticoagulant to 14 parts blood. For this step, we chose citrate phosphate dextrose. It's a standard anticoagulant used for blood preparation. It's needed for storage of the red blood cells so that they can be fresh for transfusion, and it extends the expiration time. So this is a 5-minute mixing time just to ensure that everything is well mixed and ready. So at this stage, we'll sample, make sure that all the antigens have been masked and that all the specifications have been met for this, and then the product can be bagged, labeled, and stored for transfusion at 4 degrees. So to go into some of the processing equipment, like I said, we have 3 major pieces of processing equipment. First, the blood comes from just standard transfusion bags from the Red Cross. It's going to go into a 150 liter wave bioreactor. These are purchased from GE. It's standard commercially available equipment. This works really well for us because we end up with 115 liter total working volume. So that allows for good mixing as well as a low shear environment, because basically how these work is it just rocks laterally. So that's safe for the red blood cells. The second system is a unifuge centrifugation system. This is what allows us to process the red blood cells, or the red blood cells separated from the waste continuously. So it comes with a 1.7 liter collection tank. So as that begins to fill, every 1.5 liters, the process will have to be stopped, and the blood cells will be transferred to the mixing tank. So the mixing tank is just a standard single-use disposable mixing tank. We'll use a 15 liter tank for the 10 liter working volume of red blood cells. We'll have an agitator, and all of these processes happen at room temperature, like I said, controlling the room. And then we're just going to use standard transfusion bags with proper labeling to meet all the FDA requirements for for the blood to be actually used for transfusion as a product and go into patients.
So we just talked about all our equipment, and now we're going to go into the cost surrounding this. So we're going to talk about the cost of the major equipment, the cost of the raw materials and major consumables, and then our economic conclusions. So to give you an overview, the major cost breakdown. So we have our major equipment, and that's any of our permanent pieces. We have our raw materials, which that's any of the reaction components. We're getting, like we mentioned, our blood from the Red Cross, and then for the enzyme, we're going to be outsourcing it and getting it from a commercial GMP manufacturer. And then also we're looking at the cost of the consumables, which is any of the single-use components. As for our revenue breakdown, we're going to be selling our converted blood at a price of $625 per unit. We're going to be aiming to run 5 batches a day and producing 100 units per day. And this should hopefully allow us to produce 2,500 units per year, giving us a total yearly revenue of $15.6 million. So first, for our major equipment, we have the bioreactor, the centrifuge, the mixing tank, the pump, and the welder. Together, these come to be about $500,000, and with installation costs, that's about $1.4 million. And we estimated the installation costs using a Lang factor of 3. We realized that our installation costs, we think they're a lot more than what it's going to actually be, because we have like really smaller size equipment and it's really easy to get commercially. So hopefully this overestimate should allow us to cover any necessary lab renovation and setup. So in addition to that, we have our raw materials and our consumables. And the raw materials, that's the raw blood, the PBS, the PEG, the anticoagulant, our sample kit, and then our enzyme. The two biggest costs are buying the red blood from the Red Cross, which is going to be about $5 million a year. And then our enzyme, which we're outsourcing, is going to be about $1.5 million a year. Together, all the raw materials come to be about $10 million. For our consumables, we have these single-use bags, transfusion bags, and the tubing. And those come to be about $4 million per year. And together, the raw materials and consumables are going to be about $14 million per year. As you can see, this is a lot higher than our actual equipment costs, so this is where most of our money is going for this process. So from all this, we determined that we had to sell our blood for $625 per unit, and this just allows us to be profitable. The process will generate an annual cash flow of $0.4 million a year, and we can achieve a positive net present value by the end of year 6. The 20-year internal rate of return is estimated to be 32.8%, and the total cost of production per unit of product is $602.64, leaving us with a gross profit of $22.36 per unit.
So regulatory and safety considerations are also really important for our process. Because we will be providing the blood directly to patients, we will require FDA approval, so without that, we won't be able to reach the launch phase. So in the next section, we'll be talking about some government regulations, product specifications, and we will finish with safety analysis. So our device will be classified as a Class 3 medical device. There are 3 classes for medical devices, one being the least stringent, such as things like Band-Aids, whereas Class 3 devices are those that sustain human life or are of substantial importance in preventing impairment of human health or present a potential unreasonable risk of illness or injury. So because our blood product will be supplied directly to the patients, that's why it will be rated at a higher level. And for Class 3 devices, a pre-market approval application is also required, and this is one of the most stringent requirements of the FDA. And then any product that comes in contact with blood is also regulated by the Center for Biologics Evaluation and Research, so we will be adhering to their regulatory requirements as well. And another important aspect of our clinical trials and then quality testing down the line once we launch our product are performance, functional, and interface specifications. We will have standard operating procedures for all of our equipment and also for the testing that's required with our product. And here's some of the parameters that are important for the conversion process and also for the final product. The volume and anticoagulant need to be at a certain amount in order to ensure the conversion to be complete, which is important for the safety of the product. And in addition, hemoglobin content and the A and B antigen concentration will be measured at the end of our process to make sure that the blood is safe for human use. A failure modes and effects analysis was conducted, and this is a great way to quantify the risks that are associated with the process and then sort of highlight the high priority risks. So we identified that the high severity that we predict would be from a centrifuge failure. And so a rotor failure can occur if the improper components are used in the centrifuge or the operator is using it incorrectly, maybe like imbalanced or not, if the rotor is unclean. So we recommend to conduct preventative maintenance and also use only the approved rotor for the centrifuge. And the highest occurrence that we predict would be from a burst tube connection. So as Danica described, each process is its own batch process, and then a peristaltic pump will be used to pump the fluids through tubing after sterilely welding them. So if the incorrect sterile tube welder or pump is used incorrectly, then this could result in a burst tube, which would be hazardous for our operators.
So we also performed a HAZOP analysis, which is a hazards and operability, to look at any deviations in process parameters and identify any safety concerns that can arise from that. So we were looking at the bioreactor and centrifuge and any deviations in things like flow rate, temperature, and composition. There is a risk of overflow or leak, which would cause an unsafe work environment because an operator could slip and fall and hurt themselves or be exposed to any of the compounds in the process. Also, if there's not a leak, but let's say there's a deviation in reaction composition, because enzymatic conversions require very specific parameters to react, any deviations in this could cause an incomplete reaction, and therefore our blood wouldn't be fully converted, and that would be unsafe to the patient. However, we're not too concerned about this because we do have quality assurance checkpoints at the end of the process, so we'd be able to detect this if that happened. Going back to the event of a leak and a spill, we'll have the MSDS for all 4 compounds, being the blood, the PEG, the PBS, and the anticoagulant solution in the lab. So these material and safety data sheets contain information on how to both handle a spill and what to do if there's operator exposure to any of these compounds. The 2 that are of biggest concern are the blood, which as a biologic fluid, you want to avoid direct exposure. However, we're not too worried about any of the major blood-borne diseases because we are receiving this blood from the Red Cross, meaning it should already be screened by the time that it gets to us. And then another one is the anticoagulant. Because it has citrate, which is acidic, it's especially dangerous if you get in your eye. For this, we're going to have all our operators wear proper PPE when they're in the lab, which are going to be gloves, goggles, and a lab apron.
So in summary, some of the key points that are important for our presentation today. Erythros Technologies is proposing a single-use system with a combination of enzymatic conversion and PEG masking to manufacture type O blood. We're choosing Boston for our location because of the number of trauma centers here, so there's a lot of room for opportunity. And we plan to run the process 5 times per day to produce about 10% of the daily requirements for a local hospital. Our price point is $625 per unit, and we estimate an annual cash flow of $0.4 million per year. We predict to achieve a positive net present value by the end of year 6, and the total cost of production per unit is about $603, which leaves us a gross profit per unit of $22.
So moving forward, we're always going to be looking for ways to improve our company design and our process. One of the big things we want to focus on economically is the price that we acquire blood in our enzyme. So we recognize that selling blood at $625 per unit when the Red Cross sells it for about a third of that is a problem. So we foresee that a partnership with the Red Cross could be very beneficial if we were able to acquire blood for them for cheaper and also in a specialized packaging so that we can directly put it into our process, sort of save some operation time in the initial setup. So like we said, we are going to be outsourcing our enzyme, but in the long run, if we were able to develop a GMP manufacturing production for our enzyme ourselves, that would save money. And then lastly, in terms of expansion, if we were able, we foresee the 2nd generation design of our process being a single continuous portable unit that is user-friendly and can be applied directly in the clinical setting. Looking forward towards expansion, if we were able to produce these partnered with a manufacturing company, then we were able to sell them to hospitals all over the country rather than opening one of these labs in every city. And we see that as the next step towards expansion.
So, thank you. So we have definitely some thank yous to give. Kristen, thank you so much for being such a great mentor. You really helped us out. We don't know what we would have done without you. Obviously, Professor Shane Webster, thank you guys. And then for some of our contributors, we worked really closely with a blood center at Dana-Farber, and they gave us a lot of our numbers and some facts about the blood donation process. And then also Merrimack Pharmaceuticals and Pfizer, who helped us out with some of the engineering aspects of our process. So thank you to those guys as well. And that is it. Any questions, I guess?
Questions?
I thought it was great. One, I don't know a lot about blood, so forgive me if the question is somewhat ignorant. There must be a shelf life to the blood once it's taken. I don't know what that is, but does your process affect the shelf life? I know you mentioned adding something to improve the expiration. Does that actually extend it even?
So the shelf life of blood is about 42 days after it's taken from a person. The anticoagulant is added to give it that shelf life. Okay. Our process should not affect that. It won't extend it either. So we will have to have careful expiration dates, which start from when it's pulled from the patient. So we'll need to know how old it is when we get it from the Red Cross, and our expiration will be 42 days from the beginning.
Do you have to add the anticoagulant back in after you centrifuge?
Yeah.
Yes. So just based on the density, the centrifugation step can't distinguish between the anticoagulant that was already in there. So that's the step adds it back to make sure that's back in there.
I have a question about, if you know or anybody knows, how much incompatible blood can you actually take without it being a health problem?
We do know that actually.
We
So,
do.
I mean, obviously there's like little kids and adults and like everyone's body weight ranges, but like for a typical, like I guess, adult, it's 50 mils that can be transfused into.
Okay.
And like, you're not gonna be feeling good after that, but it's not gonna cause any like super lethal reactions. So that's why we can't claim our process is gonna give you 100% conversion. We can say that it's gonna give, high enough, we're gonna meet those specs.
Yeah.
So I definitely don't think there's gonna be 50 mils left in there, but that is the max you can receive.
Yeah, and that makes and I know you have a good section on quality control, but that makes that even more important.
Yeah.
Right.
So that would be a consideration as well. With our specification is 2% antigen, so that calculation would be carried out to maximize the 50 mils, because often you receive more than one unit of blood, so that would have to be carried through.
How much was your liability insurance policy?
I think we calculated the insurance.
Oh, we got a number from Shelby.
Well,
For-
no, we got intellectual property and.
I'm thinking of like product liability.
Like,
Yeah,
if
it's
a
a
patient were
good
to see
question.
this and
Well,
So that would be
Something goes wrong.
significant.
Protects you.
Yeah.
keeps you busy.
Yeah.
The other thing, the dynamic, the way blood is collected and distributed. Do you have any sense at all in blood usage, how much is demanded because of trauma versus volunteer or sort of thing?
Yeah, I got those numbers. I work in a blood bank. I just specifically asked them how much they were required to apply to the hospital that they provide. They didn't break it down into, this is the amount we need for traumas, this is the amount we just need for like someone who's on chemotherapy or something.
Well, I'm thinking of voluntary surgeries that can be delayed. There's nothing critical.
So that was a consideration with us supplying only 10% of the total volume, because like you said, in situations where it's not an emergency, the blood can be typed. And people, like, if you're type A, you can be given safely type A blood. So we don't want to be supplying 100% of the converted blood, because.
And being in a city with 9 trauma centers,
Yeah.
we definitely think that with our production goal, there would be enough of a demand.
I would have also just said, I think a business model that might have made more sense to me would be, instead of trying to sell to the hospitals, where you don't know what they have in inventory, you don't know what their suppliers have in inventory, you don't know what they're demanding at any one point in time, is sell it back, do a conversion and sell it back to Red Cross or to other buildings. Because that's who the hospitals are in touch with when they have demands.
And that's kind of like our recommendations. We were thinking, partner with the Red Cross and figure out something like that.
Definitely something to consider.
Any last questions? Excellent, very nice.
Thank you guys for listening.